I called it computational biology, and then you mentioned that more recently it's being called biomedical data science. Tell us about your particular area of research.
Yeah, so I guess it's fairly broad, but at the core of it is how I think about protein structure. So this is the three-dimensional structure of proteins and DNA and RNA for that matter. But I focus particularly on proteins. And we were involved in helping develop a resource called the Protein Data Bank, which is the public repository, the open source, the open data repository for all of this material that has been collected now over my lifetime. When I started, there were 77 structures within this data resource. There's now close to 200,000. And that's had a profound effect on how we understand biology. My research has involved a lot around that, including developing algorithms that compare structures with each other, actually find binding sites within these structures such that they can be used to actually design drugs. It's been, and that those are two areas. We also used structure. My favorite one is actually how we've used structure to study evolution. It's just, I'll bore you with just an example of that for 1 second. Is
Please.
that it's
There's
going to
nothing
happen
the
anyway.
listeners love more than being bored by intricate data science details. No, and I'm serious. They will never be bored. This is what we're here for. So hit us.
So if you think about the data of proteins, a protein consists of about 300 amino acids, and there are 20 different types of amino acid. That is 20. So if you took all the random possibilities for proteins, that's 20 to the 300. That is more than all of the atoms in the universe. So why is nature only, we know several million of these, but that, and there's probably certainly more to be discovered, but the number is extremely small. So nature has basically given this incredible reductionism in creating life. Then what's even more profound is that of those millions, they all fold up into a matter of 1,000 three-dimensional structures. So what is that? So that was something that I thought was just incredible. Nature's done this, and there's like several thousand jigsaw pieces that when you put them together in different ways, make up everything that, every species. So what is, you start thinking to yourself, what does that mean? And it means, one of the things it means is to create a new jigsaw piece, to create a new three-dimensional shape is actually a pretty rare event. And so we actually started looking at now, with data, there's enough data that you can actually see the shapes that exist in many, many different thousands of species. And so one afternoon, just on a laptop, we did an experiment where we took that data and we basically said, so you could say, okay, I have all these shapes on one axis, and then on the other axis, I have every different species that has those shapes. And it's a matrix. And in each cell of that matrix, if that shape exists in that species, I give it a 1. If it doesn't exist, I give it a 0. That binary matrix can be converted into a tree. That tree is the tree of life. So it's just stunning that in an afternoon on a laptop, you could actually do what many evolutionary biologists have spent inordinate amounts of time doing, even before doing it with. You can do it with sequences as well, but this is. I just got an amazing kick out of that, and if that's not boring you enough, I'll take it one step further.
Please, I'm so desperately bored. I need more.
So when we were publishing a paper that described this, I actually gave it to my bioinformatics class at UCSD as the final exam. This is double dipping. So basically, this is your final exam. I want you to propose the next set of experiments based on this paper. What would you do next? And, with the hope that, of course, I got a lot of rubbish, but I also got, there was a student who was actually at Scripps Institute of Oceanography, and he
Mhm.
pointed out to me that, I kind of knew it, but he highlighted it, that 90-something% of all evolution has occurred in the ocean based on the 4 billion years of the Earth's existence.
Oh, wow. I didn't know
So,
that.
and he was an oceanographer. And he said, and in that time, the geochemistry of the ocean has changed dramatically, particularly from what's called reductive to eugenic to oxidative. And he said, I wonder whether we can see those fingerprints of those changes in these structures that we're talking
Oh.
about. And lo and behold, we found amazing correlations between the changes that have occurred in the geochemistry of the ocean because of that change from reductive to oxidative. That shaped life. So gets back to why data science is so important, because basically you've had a whole bunch of people who study geochemistry, you have a whole bunch of people who study evolution, but you've had very, very few people who ever studied both. And suddenly the
Right.
data brings them together. Data can bring them together, and you start making new discoveries about ourselves, about life itself. So in the grand scheme of academia and scholarship, that didn't get me the most citations, but it's actually the work I'm in some ways that we're most proud of.
That was such a cool example. And it is mind blowing to me to think. So, you probably don't know this about me, but I have a biology background. So, my PhD is in neuroscience, and I did a lot of biology, particularly as an undergrad, including evolutionary biology courses. And so, I know things about genomics and about proteomics, but I did not know this fact that there's only a few thousand kinds of protein shapes, functional protein shapes, and that very small number. Because I often think in my mind that there must be orders of magnitude more of that, given the diversity of function that different organisms can have. It's wild to me that from a plant, a single celled organism like an amoeba, and me, and a leopard, and a turtle, that you can get these wildly divergent organisms from just a few thousands of protein structures. That's really cool.
Yeah, it is, and I think it's my favorite fact in all of biology that there's that level of reductionism, and of course, they come together in many different ways, to form. I mean, this happens at actually what you would call the domain level or at the fold level. So there's lots of nuances about this, but the general principle is correct.
So and a thought that occurs to me, given the increasing level of understanding that we have of protein structure, could we then engineer new protein structures um, to perform some kind of function that maybe doesn't happen in nature? So, could we be engineering protein structures to allow us to clean up the oceans, or to convert to fix carbon dioxide from the atmosphere into a fuel that we could be burning? Have bacteria that are doing that? Should we? So this is quite an open-ended question, and it wasn't one I was anticipating asking. So I know you weren't prepared for it, but do we have, if we're looking to have these new kinds of functions, if we want to engineer bacteria or some other organism to be doing something functional for us, should we be looking at the thousands of protein structures that already exist and recombining those in new ways? Or could we be thinking about new kinds of protein function that don't exist in nature?
Well, remember I said that. Well, the short answer is yes. Um, but remember I said that there's millions of sequences and then there are thousands of structures. So,
Right.
what's going on now, and this really, it comes right back to data science, as you know, that AlphaFold 2 was this development by DeepMind, a spin-off from Google,
Mhm.
to actually predict the structures from those sequences. This was the science's breakthrough of the year last year, and
Mhm.
it's actually a profound development. So what's happening now as we speak might actually be what's done, is to actually predict the structure of all of those millions. So instead of having, of course, when we do that, question is, are there things that come out which are new that are not encompassed by this in the thousands? And
Right.
and then that then gives us a bigger picture of what protein fold space looks like. Right? And then the question is, well, where are the gaps in that space? There's clearly going to be gaps between things that we already know, and then there are things that are outliers that we have no idea. So yes, there is the prince, and this kind of protein synthesis is clearly the next frontier. And it's sort of reverse engineering, right? You've got these where do you start from? You engineer a sequence that then is going to give you a new structure. So there's clearly going to be movement in that direction to address exactly the kind of issues that you're talking about. But there's again, like all of these things, it's easy to sit here at a podcast and talk about it for a few minutes, but actually there's a lot of sweat and tears to actually make these things happen. But the potential is there,